Good morning, good afternoon, everyone. My name is Selwyn Ho, and I'm the CEO of Medigene, and I have the pleasure of welcoming you to today's R&D event. The focus for today's presentations are to perform a deep dive into Medigene's core expertise in discovering and developing what we believe are extraordinary TCRs or T-cell receptors for a range of TCR-guided modalities. Our agenda today will aim to cover off a number of topics related to TCRs, which we hope will provide context on our current strategy and the research underpinning this. Firstly, from our Chief Scientific Officer, Dolores Schendel, why extraordinary TCRs are ideal cancer antigen binders, and why future TCR T-cell engagers or TCR-TCEs will deliver on their potential. Next, Barbara Lush, our Head of Technology and Innovation, will cover a new set of innovations within our E2E platform that has potential broad applicability. Following this, we are delighted to have Dr. David Zhen from the Fred Hutch Cancer Center in Seattle, who will talk about our lead program, MDG 1015, and how he sees the potential value for MDG 1015 as a therapeutic agent for his patients. Finally, Kirsty Krame, our Head of Clinical Development, will summarize and discuss our current pipeline and data catalysts. Following the formal presentations, there will be time for Q&A, which we will take on a first-come, first-served basis. We will be making a number of forward-looking statements during this event, and I draw your attention to this slide, which can also be found on our website, medigene.com. Medigene is a clinic-ready stage oncology platform company developing multiple TCR-guided immunotherapies. We have a highly experienced leadership team with multi-decade scientific and biopharmaceutical experience. The basis for our belief in our highly differentiated, extraordinary T-cell receptors and potential for highly differentiated TCR-guided modalities is our end-to-end platform. We believe that we have established clear scientific differentiation of our TCRs and platform technologies through our partnerships with BioNTech, Regeneron, and most recently with WuXi Biologics. Our E2E platform has delivered a focused pipeline versus validated cancer antigens, both cancer testis antigens and neoantigens, where based on the TCRs that we have announced, this could target approximately 500,000 patients globally on an annual basis. I won't discuss this in more depth now, as both Dolores and David will discuss this later on for our KRAS target discovery, our lead program MDG 1015, and our recent move into TCR T-cell engagers with MDG 3010. Finally, the programs give us the potential for a number of near-term data catalysts, including initial data from the dose escalation phase 1 clinical trial from MDG 1015, as well as progress on our KRAS and MDG 3010 programs. Our vision as a company is to discover and generate a library of multiple, extraordinary 3S or safe, sensitive, and specific T-cell receptors that we can then deploy across multiple modalities, including autologous TCR-T therapies, allogeneic cell therapies, and TCR T-cell engagers. We will both develop and partner these TCRs and TCR-guided modalities, aiming to achieve clinical validation of these modalities ourselves or through our partners. We will also advance and innovate our E2E platform, and where appropriate, to partner out certain technologies that could be of benefit. Ultimately, we hope that these therapies fulfill their potential and become available to patients. We believe that TCR-guided modalities offer broad potential as part of a portfolio of next-generation precision therapies. On the left-hand side of this slide, we show that as understanding into cancer treatments has advanced, it has been well accepted that there is the need to have different options to target multiple pathways that underpin cancer development, either through therapies that enhance or activate the immune system, or directly kill tumor cells. We believe that in either of these scenarios, TCR-guided modalities, through either autologous or off-the-shelf modalities, can play a key part within the treatment paradigm. Currently, Medigene has a pipeline where we have developed our 3S TCRs across both autologous TCR T-cell therapies that we are developing in-house such as MDG1015, as well as through our partners BioNTech and Regeneron. In addition, with our partner WuXi Biologics, we are developing MDG3010, our TCR T-cell engager. We will discuss MDG1015 and MDG3010 in more depth later on, and Kirsty will also discuss our current estimates for our data catalysts. So with that, I'm delighted to hand over to Dolores, who will walk us through why extraordinary TCRs are ideal cancer antigen binders.
Thank you, Selvin. It's my pleasure to discuss the science and technologies behind our immunotherapies. We developed 3 TCR-guided modalities. 2 are cell-based therapies in the form of autologous TCR T therapies prepared for individual patients, or allogeneic TCR-expressing natural killer cells as off-the-shelf cellular therapies. The third uses soluble TCRs as TCR T-cell engagers, also as off-the-shelf therapies. All 3 employ our special 3S TCRs to guide effector cells to tumor cells and trigger tumor cell destruction. This mix provides treatment options for patients at different stages of disease. I'll describe our work on the first and third modalities and leave you in suspense about our TCR-NK cells, but we'll reveal more about this platform in the near future. Our immunotherapies are rooted in technologies that we've assembled over the past decade in our modular and dynamic end-to-end platform. In this platform, we combine multiple exclusive and proprietary technologies to create our cutting-edge immunotherapies for solid cancer. TCR selection is the first component and the critical element for our immunotherapies. So starting at the left, through target screening, we choose a target antigen and then use our TCR generation and optimization module to discover TCRs for that specificity. The next 2 modules are used to make best-in-class cellular therapies with armoring and enhancement technologies, including our co-stimulatory switch proteins. Equally important is our module for optimal drug product manufacturing. And at the end, additional technologies support our clinical development and can be modified for different TCR-guided therapies. Innovations are added to each module, and they come in 3 flavors: safety enhancements, shown in yellow, efficacy enhancements in red, and development optimizations in green for faster, better, and cheaper design and development of TCR-guided therapeutics. So let's examine some details about Medigene's 3S TCRs that move them from being ordinary to become extraordinary. Here is a side-by-side comparison of our 3S TCRs versus antibodies, both serving as binders to guide cellular therapies or T-cell engagers to tumor cells. At a glance, you easily see that many attributes are only found with 3S TCRs. I'll consider a few here, and others will be covered as we look at some upcoming data. We developed 3S TCRs that are truly tumor-specific through selection of the target antigens in the form of short peptides that can be derived from the entire proteome. For example, we use tumor-specific mutations or cancer testis antigens that are rarely found in healthy tissues. These short peptides are bound to HLA molecules in a complex that is presented at the tumor cell surface. This elegant mechanism allows T cells to recognize aberrant intracellular proteins that are not accessible to antibodies. These peptides are picked up inside the tumor cell and transported by HLA molecules to the cell surface, where they can be seen by TCRs. In contrast, target antigen selection and tumor specificity is far more constrained for antibody binders, since they only bind 3-dimensional cell surface molecules, which represent about 10% of the proteome. We also select natural TCR sequences that do not need to be mutated to display exceptional sensitivity for tumor cell recognition. This brings another level of safety, since the TCRs were present in healthy donors. Antibodies with precisely defined specificities can be used for CAR Ts and T-cell engagers, but their targets are often found on healthy tissues. A clear example of this is the CD19 CAR T-cells that recognize healthy B cells. The highly differentiated safety profile of 3S TCRs reduces chances for side effects and allows higher dosing with potential for greater clinical efficacy. Antibody-directed immunotherapies face a major hurdle that also constrains their target specificities. Many antibody-directed target antigens can be shed from the tumor cell surface through cleavage of an extracellular domain that's released into the circulation. Shed ectodomains can prevent recognition and destruction of tumor cells by direct target loss from the tumor cell surface, or they can build a reservoir of antigen in the bloodstream that inhibits the drug product from reaching the tumor cell. While this is a major problem for antibody-directed immunotherapies, fortunately, it's not relevant at all for TCR-guided immunotherapies. The TCR target is strongly anchored on the tumor cell surface via peptide HLA complex, and that is not subject to cleavage and release of shed antigen, bypassing this key challenge for antibody binders. However, TCR-guided therapies must overcome other challenges. HLA restriction allows targeting of many intracellular proteins and brings additional safety in this context. But HLA is very diverse, and one HLA does not fit all patients. However, combination of several allotypes, as shown on the left, can cover large fractions of the world population. On the right, target antigen expression can vary among tumors of different types and for different patients, and this requires careful indication and patient selection for any TCR-based immunotherapy. Implementation of TCR multiplexing is our direct pathway to overcome these hurdles. As Selvin showed in our pipeline, Medigene and its partners developed different 3S TCRs which are discovered at Medigene and can contribute to solving heterogeneity of both types. We explore TCRs specific for different antigens presented by one HLA allotype to enable different TCRs to be multiplexed to combat tumor antigen heterogeneity. TCRs restricted by different HLAs that present different peptides that are derived from the same target antigen can address HLA population diversity. TCRs seeing peptides presented by as few as 5 different HLA allotypes can rapidly expand the patients that can be treated despite high population diversity. So 3S TCRs are clearly the choice of preference for different types of cancer, but they must be extraordinary TCRs. And I'd like to demonstrate how Medigene's platform is uniquely positioned to find and develop such extraordinary 3S TCRs, irrespective of their target specificity or HLA restriction. We employ a de novo priming approach to stimulate T cells of young, healthy donors to acquire TCR candidates in a rapid, high-throughput screening process. We collect TCRs from different donors, and we build a basket of novel, natural, high-affinity TCRs, each having a unique sequence that will impact the 3S characteristics of specificity, sensitivity, and safety. This natural sequence variability allows us to select optimal TCRs with high sensitivity for tumor recognition and no signs of cross-reactivity for healthy tissues. Our TCRs do not need to be mutated to improve their sensitivity, giving them a better safety profile. By using healthy donors rather than patients, we bypass constraints in access to high-quality starting materials for T-cell priming. Patient cells often show limited proliferation, poor function, and restricted T-cell receptor diversity through long-term exposure to tumor immunosuppression and tumor editing, limiting the chances for successful TCR isolation. Let's put some numbers around our TCR discovery approach. For two TCRs, we've recently added to Medigene's pipeline. KRAS mutation-specific TCRs are a special focus for us, but they're really challenging to find, since they must distinguish single amino acid differences between mutant and ubiquitous wild-type KRAS protein. We used 20 donors and characterized 50,000 individual T-cell clones, recognizing G12V presented by HLA-11. These numbers were dramatically reduced by testing functional recognition of tumor cells with low antigen levels. In the end, only 6 candidates survived our first-level selection process. 27 donors and 70,000 clones were screened to yield 9 unique sequences for G12D specific TCRs. In the end, we have a small batch of TCRs, despite starting with tens of thousands of antigen-specific living T-cell clones. Based on presentations of our competitors, it does not seem that they employ the breadth and depth we use for TCR discovery, perhaps only considering 100 or fewer starting T-cells and TCRs. Finding TCRs with even a 5 to 10-fold higher sensitivity can make a substantial difference in recognition of tumors with very low levels of antigen, but these are very rare. We invest the time and energy needed to find these very rare and very exceptional TCRs, which cross the boundary from ordinary to extraordinary. So I'd like to take some minutes to look at exemplary data for our G12D specific 3S TCR, TCR-16, to illustrate the outcome of our searches. In all experiments, I'll show you TCR T-cells made from 3 independent donors to show consistency of responses. These are modified to express TCR-16 or left as an unmodified negative control. Looking at specificity, you see that TCR-16 only responds to G12D peptide presented by HLA-11 positive antigen-presenting cells, which are pulsed with the exogenous peptide on the left, or loaded with RNA that is processed and presented as peptide by the cellular machinery on the right. TCR-16 shows no recognition of B, C, or R mutated peptides, nor any response to wild-type KRAS peptide. This demonstrates the remarkable single amino acid specificity of TCR-16 for G12D peptide. This exquisite specificity of TCR-16 is coupled to very high sensitivity, detected by cytokine secretion in response to graded amounts of G12D peptide, with a half maximal response in the picomolar range, indicated by the dashed lines for all 3 donors. Normally, very high sensitivity can lead to loss of exquisite specificity, but we can find TCRs like TCR-16 where this is not the case due to our extensive searches. On the right, T cells with TCR-16 efficiently kill tumor cells expressing natural levels of endogenous G12D antigen, while no killing is seen for tumor cells with wild-type KRAS protein, which is a critical safety control. Safety is of utmost importance, and multiple assessments ensure that TCR-16 does not recognize healthy tissues. Here is an example of 8 healthy tissue types taken from our larger test panel. Concentrate on the cardiomyocytes at the left. There are 12 columns, 9 of which are barely visible. The first 3 columns are the most important. These are T cells from the 3 donors with TCR-16, and they do not see the heart cells. The next 3 columns are expected to be negative, as these are T cells that do not express TCR-16. Next come the 3 positive control T cells that respond strongly to cardiomyocytes pulsed with G12D peptide. Finally, the last 3 are negative control T cells without TCR-16 that do not respond to peptide pulsed cardiomyocytes. Similar patterns are seen for all 8 healthy cell lines. They can be recognized by TCR-16, but only after pulsing with G12D peptide. From these data, we can conclude that healthy cells are not recognized in their natural state by TCR-16, demonstrating that TCR-16 does not show any off-target toxicity for healthy cells. Our proprietary vetting algorithms employ multiple assays to assess each 3S attribute. I've shown you one example each for the 3S characteristics of TCR-16. These assessments assure us that our lead TCRs have exquisite specificity, high sensitivity to see tumor cells with very low levels of antigen, and have a highly favorable safety profile. They do not display any on-target, off-tumor, or off-target toxicity for healthy tissues. They must pass our stringent criteria for further clinical development. We also determine if TCRs are CD8 co-receptor independent. If so, they can be used directly in CD4 T cells that do not naturally express CD8. So now I would like to turn to our TCR engager program using 3S TCRs that are isolated and characterized just as I've shown here for the TCR T-cells. TCR T-cell engagers are new TCR-guided modality recently incorporated in our immunotherapy programs through our announced co-development alliance with WuXi Biologics. Antibody-derived T-cell engagers have been extensively studied in the field and revealed to have several impediments for optimal clinical outcomes. T-cell engagers have two arms. One binds to a target antigen on tumor cells, and the second binds to T cells, normally via surface CD3. This builds a bridge between the T cell and the tumor cell, bringing them into close proximity and allowing the T cell that's activated by engager binding to CD3 to attack the tumor cells. While this is a simple concept, there are numerous hurdles that impact efficacy, safety, and durability of outcomes of patient treatment. If we start with number 1, looking at the tumor binding arm of the engager, there are major deficits for antibody targets that differentiate tumor cells and healthy tissues, as we've already discussed. Engager binding healthy tissues can lead to nonspecific activation of T cells and high toxicity. Hopefully, you can immediately understand why 3S TCRs that can precisely differentiate tumors from healthy tissues can elegantly overcome this hurdle. Looking at number 2, the CD3 binding arm, we know that the affinity and kinetics of engagement of CD3 with T cells controls the level and duration of T cell activation. If the affinity of this CD3 binder is too high or duration of binding is too long, the T cells can be driven to exhaustion and even cell death. If the affinity is too low or too short in duration, the engager will have a lesser impact on tumor destruction. So both parameters will impact clinical efficacy. With respect to 3, the structure of the engager is critical. Some engagers lack FC regions. They may penetrate tumor cells more easily, but they require continuous infusion with a dramatic lowering of quality of life for patients under treatment. Lack of FC receptors, or with purposely impaired functions, can limit potency through failure to recruit secondary immune responses. Finally, as point 4, tumors with poor T cell infiltrates or insufficient T cell functionality will not respond well, despite using an excellent engager design. Selection of patients, indications, and line of treatment is critical for clinical success. The MDG 3010 program aims to overcome many hurdles and provide best-in-class TCR-guided engagers. Medigene and WuXi Biologics have joined forces to bring together our highly complementary technologies to solve several critical impediments just described. WuXi Bio's well-known proprietary WuXi body platform has overcome many of the challenges for antibody-based engagers, and their approaches are implemented in more than 40 different programs. Expanding on this extensive expertise, WuXi Bio has built the TCR body platform to provide a proprietary and stable scaffold for TCR T-cell engagers. This platform allows efficient comparison of TCR binders with easy improvement in binding affinities if required. It allows soluble TCR expression to be easily studied and the best scaffolds to be selected based on individual TCR sequences. Collaborations with pharma validate the importance of this platform. It will allow Medigene and WuXi to co-develop the TCR targeting arm of the engagers, taking advantage of TCR body scaffolds and Medigene 3S TCRs. WuXi Bio has optimized their anti-CD3 binding arm to allow activated T cells to kill tumor cells without undergoing exhaustion. This proprietary CD3 binder demonstrates a fast on and fast off rate, shown in the upper left. This avoids overstimulation of T cells, as may occur with binders that remain attached to the T cells for a longer time, shown with the benchmark engager shown in the lower left. The optimized CD3 binders provide signals to T cells that activate greater levels of killing activity in the upper right in red, and lower levels of cytokine release in the lower right, as compared to the benchmark binder in blue. This optimized CD3 arm should contribute to greater T cell functionality and persistence, and lower toxicity caused by cytokine release syndrome in patients from overshooting T cell activation. This improved CD3 binder is already in clinical phase 1 studies with 4 different TCR T-cell engagers. In summary, the combined technology platforms of Medigene and WuXi Bio allow an optimal 3S TCR binder to be coupled with an optimized CD3 binder and the best structural format for each TCR engager to be efficiently chosen. Adding in targeted selection of patients and indications, 3S-guided TCR engagers open the door to provide patients in the future with superior treatment outcomes. This is a very exciting development for Medigene that taps immediately into the 3S TCRs we've discovered in the past and efficiently applies our E2E platform technologies to this new off-the-shelf TCR-guided immunotherapy. We see a great future for Medigene and for our patients by using our deep know-how to develop these new TCR-guided immunotherapies. This now completes my points of discussion, and I'd like to leave you with one thought. If George Orwell were an immunologist working at Medigene today, he would certainly say that all TCRs are equal, but some are more equal than others. And this is the basis for our claim that we make the ordinary TCRs extraordinary. And now I hand over to Barbara Lusch, who will present an exciting innovation recently added to the E2E platform.
Thank you, Dolores. So it's my pleasure to introduce our latest invention, a universal tagging and tracking system for TCRs called UniTope and TraCR. Let me first outline the benefits and drawbacks of those methods that are most commonly used both in R&D and in clinical settings. On the left side, you see peptide HLA multimers, which target the TCR antigen binding site and are unique for each specific TCR. They often bind to TCRs of other specificity, which must be carefully assessed and controlled. And it is difficult and sometimes even impossible to create multimers for some peptide HLA ligands. These reagents are then very labile and with a very short shelf life. In the middle, we find the especially in R&D most commonly used tracking system, the TCR variable beta-specific monoclonal antibodies, which are very reliable and have a long shelf life. But they bind to both the recombinant TCR and to other T cells in the natural repertoire of the patient that use the same V-beta chain. So to properly enumerate TCR T-cells, one has to determine background levels, meaning for each patient, untransduced T-cells must be analyzed from the same starting materials. And there's increasing evidence that this might be problematic in regulatory developments in the future. On the right, you find tag-specific monoclonal antibodies. They bind to an artificial epitope expressed on the TCR T-cell surface, and this tag is encoded as a separate gene, and thereby it's only suited for use to detect TCR T-cells that co-express both genes, the TCR and the tag. Highly concordant co-expression is required, but not always achieved for accurate enumeration of TCR T-cells. Given the limits of the existing tools, we developed a new, exclusive and proprietary tagging and tracking system that combines all the advantages of the methods shown so far, but at the same time eliminates their weaknesses for faster, better, and cheaper design and development of TCR-guided therapeutics. On this slide, you see the characteristics of UniTope and TraCR outlined. UniTope is a short epitope of only 6 amino acids, allowing it to be integrated into the TCR itself, ensuring 100% co-expression without disturbing the functionality of the TCR. Since this epitope is highly homologous to human sequences, we expect no or only minor immunogenicity. The TraCR antibody shows a long shelf life, but what's more important, it's specific for UniTope, meaning it does not bind to unmodified TCR T-cells and thereby eliminating the need to compare the final product with untransduced T-cells from the same starting material. These attributes render UniTope and TraCR applicable for highly standardized tracking and enrichment of TCR T-cells. Additionally, the system comes with remarkable time and cost savings. Now, allow me to go more into detail. The biggest hurdle in UniTope development was the integration of the epitope in the TCR without disturbing its functionality. We performed intensive 3D modeling. One of the TCR models is depicted on the left side with the alpha chain in blue and the beta chain in gold. With these models, we could identify 3 loops, 2 in the beta constant chain and 1 in the alpha constant chain, that facilitate integration of UniTope without disturbing the TCR's intra- and intermolecular interactions and in last consequence its functionality. To prove those statements, the next slide shows representative experimental data for one of the many identified positions in one of several tested TCRs. Please first focus on the left side. Here we analyzed T cells that were mock transfused, left column, transduced with an unmodified TCR in the middle, or transduced with the UniTope tagged TCRs on the right. In the lower row, staining with a commercially available antibody specific for the beta variable chain is shown. We retrieved an equal staining of the unmodified as well as for the UniTope tagged TCRs. These are the middle and right box in the lower row. And this certifies that UniTope does not interfere with the expression of the TCR. The upper panel shows the staining of the same cells with TraCR. TraCR antibody specifically binds only to the UniTope tagged TCR T-cells given in the upper right box, and this staining is equally good as for the VV-beta antibody in the lower right box, and therefore we can state that TraCR can substitute for the V-beta specific antibody. On the right, the results of a functional assay are depicted. T-cells equipped with either an unmodified TCR or a UniTope-modified TCR were co-cultures with peptide-loaded target cells, and the interferon gamma production was measured by ELISA. If you focus on the pink bars, you see that both types of TCR T-cells produce equal amounts of the cytokine, proving that integration of UniTope does not interfere with functionality. For the sake of time, I cannot show you all experiments performed to in-depth characterize UniTope and TraCR. Therefore, the next slide summarizes our efforts. We compared UniTope-equipped TCRs with 2 non-modified TCRs. By this, we could prove that the insertion of UniTope does not influence 3 S characteristics. For safety, specificity, and sensitivity, no significant differences could be identified in any of the performed experiments. As Dolores pointed out, safety is of the utmost importance for us. Therefore, I want to highlight that we tested 16 healthy cell types and over 300 mismatched peptides, and no significant deviation was detected between modified and unmodified TCRs. The next slide summarizes the attributes and benefits of our high-precision technology. Seamless integration of a short epitope directly into the TCR sequence ensures 100% co-expression, and this makes it easy to adapt even to complex gene transfer systems. More importantly, it's also suited for drug product development and quality control, enabling precise drug dosing without the need to determine background levels from unmodified starting material from the same patient. And also, follow-up pharmacokinetic studies in patients receiving recombinant TCR-T therapies is possible. GMP-grade TraCR allows the enrichment of the product, if this is needed, and the humanized version of TraCR will be suitable for in vivo tracking of the UniTope tagged TCR T-cells. Since the system has to be developed and validated only once and can be used afterwards for every TCR of interest, UniTope and TraCR significantly simplifies the regulatory development of TCR products. And we are so excited about this new technology, which answers so many needs in the CMC fields, that we're incorporating this new technology for faster, better, and cheaper design and development of TCR-guided therapeutics in our platform. And with this, I would like to hand over to Dr. David Zhen, who will present details of our upcoming clinical trial.
Hello, everyone. First, I would like to thank the Medigene team for the invitation to speak, as well as to be the lead investigator and at having the Fred Hutch lead the study efforts, which we're very excited about. So we'll start with some preclinical data in terms of the design for the epitome study. So this slide summarizes essentially the TCR-T therapies that are under investigation and approved. And these are what we've referred to as the first and second generation TCR-Ts. So as of now, we have one FDA-approved TCR-T therapy, that's Tecelera for essentially synovial sarcoma. And while there have been advancements, and it's very exciting that we have such improved therapies, the relative benefit of such therapies remain limited. As you can see here, the median progression-free survival is around 4 months. Median overall survival is around 16.9 months. And one of the issues in terms of the limitations of the current first and second generation TCR-T therapies is overcoming a very robust immunosuppressive immune microenvironment. And secondly, what you can notice here is there's a concerning trend in terms of toxicity rates from CRS and ICANS toxicities, which could be contributed by the fact that many of these therapies contain CD4 positive T cells within their product, which can be contributing to their toxicities. And so really when we think about the next generation is how do we overcome the tumor microenvironment and also improve the products to reduce the CD4 count and improve toxicity. So this is where Medigene's platform is very novel, that this third generation TCR-T is hoping to overcome the unmet needs of first and second generation TCR-T therapies. And we'll go through the unique aspects of this construct. So first, in the first and second generation TCR-T therapies, the TCR requires a mutation to enhance affinity. Medigene's TCR-T therapies, however, does not rely on the mutations, which then increases the safety profile. For the second component that's important is that there is a costimulatory switch protein, which targets PD-1 with a 4-1BB stimulatory domain, which the idea here is to overcome the immunosuppressive microenvironment, further enhancing activity. And this is strictly gated with the TCR. And finally, the last aspect of this TCR construct compared to first and second generations is enriching basically for CD8 positive T cells and limiting the amount of CD4 product. And this is done by a rapid cell production with a 20-day vein-to-vein time. And the idea here is that with a quick production that we enrich for younger and fitter cells and enriching for CD8 cells, further enhancing the activity and increasing the safety profile. So the MDMG1015 is a optimal affinity TCR. And as mentioned earlier, the unique aspect is compared to other TCR TCR T therapies, is this is a natural unenhanced naked TCR, and it does not require mutations for affinity maturation. So this is an experiment demonstrating multiple different cell lines where we can have good recognition and activity of this naked TCR, even at very low levels of target MRA. And again, this is compared to a benchmark TCR, which is a GSK affinity matured TCR. Furthermore, in demonstrating activity, this is a synovial sarcoma cell line, SW9A2, and this is to highlight the activity of incorporating this PD-141BB costimulatory switch protein, the CSP. What you can see here is in the gray lines is that while there is activity with a recognizing antigen without the CSP, when we add the CSP to the TCR-T construct, we have 2.5 times the cell proliferation with this, and this does not require CD4 T cell activity to see with this. And this is further demonstrated in this culture cell model where we look at various different tumor cells, and what we can see here is demonstrating that the PD-1 and 41BB CSP is gated to the TCR, and we can see rapid and sustained in vitro tumor cell killing upon serial rechallenges. And what you can see here is in the top row is that basically cells that either express the antigen, in this case NY-ESO-1, as well as PD-1, have rapid and sustained killing. You'll see in the second panel is a different cell line, which demonstrates, as compared to a TCR without the CSP, that it can target cells that express the antigen, but will still have activity without PD-1. But I think what's very important is in the third panel here is that this CSP is totally gated with the TCR, and that we do not have any cell killing when the antigen is not expressed. And this is further highlighted in a co-culture here, where we're looking at two different types of cell lines, tumor cells. One is a melanoma cell line, which is antigen expressing, but also PD-1 expressing, and a urothelial carcinoma cell line here expressed in green, which is expresses PD-1 only, but does not express the antigen. And I think what's very impressive here is that there is no innocent bystander killing with this, again, demonstrating the strict gating of this TCR with this TSP. And likewise with this, this construct with a naked TCR allows for the development of polyfunctional T cells, which produce multiple cytokines, which enhances an effective immune response without the need for CD4 positive T cells. And in fact, there's an 8 times increase in single cytokine production. And this is not just in terms of the number of cells that increase their cytokine production, but on a single cell level, each of the cells can also increase the repertoire of the type of cytokines that can be produced. And as we've mentioned, really the goal here with this is that we're enriching for a CD8 positive T cell product in the absence of CD4 Treg control mechanisms, with the idea that we can increase and maximize the benefit in treatment of solid tumors. And likewise, also with the hope that we can also decrease the toxicity that has been seen with first and second generation TCR-T therapies, including decreasing the risk of cytokine release syndrome and as well as other toxicities. And with this, I think this is actually very important for me as a clinician, and I've been involved in several different adoptive cellular therapies, is the short manufacturing time. So one of the limitations as we've been treating patients with solid tumors is the long manufacturing times, often several months required for production. And often patients with solid tumors have very limited prognosis, and they can progress very quickly. And I think what is novel about this construct is a very short manufacturing time, where 6 days is kind of the optimal time period. And this limited manufacturing time is hopefully enriching for CD8 cells, as well as increasing the proportion of T memory cells, which further enhances potentially the clinical efficacy. And we can see up to 85 to 95% of T memory cells with this product, as compared to current commercial products, which only reach about 18% at best. And so we'll go into and jump into the study design for Epitome 1015. One, which is a phase 1 clinical trial. And so the primary endpoint for the study is to establish the RPT, the recommended phase 2 dose of MDG 1015 on the safety, tolerability, and dose-limiting toxicities, and also evaluating for preliminary efficacy. And the secondary endpoints is assessing the feasibility of this third-generation product in a specific solid tumor patient population, and we'll be further assessing safety and tolerability, and as well as efficacy, as well as other PK endpoints. So in the EPITOME-1 study, we will start with a dose escalation design, and we'll be evaluating several different dose levels with also an accelerated dose phase such that we can move on to the dose expansion quickly if we don't see toxicities. But if we do, there will be a more traditional 3+3 design that will be incorporated. And we will be evaluating patients with a variety of different solid tumors, including synovial sarcoma, myxoid round cell liposarcoma, ovarian cancer, and also gastroesophageal adenocarcinoma. And the reason these cancers are being chosen is these are the cancers that do express NY-ESO-1. Upon determining safety, we'll move on to dose expansion with individual cohorts at the recommended phase 2 dose with the idea of further identifying the clinical efficacy. And this is really a rough study schema. I know a little bit hard to see the tables on the bottom, but the tables on the bottom are really giving a highlights of the key inclusion exclusion criteria. Again, these will be patients with the refractory solid tumors that I mentioned. Patients must have an HLA-A201 expression, as well as NY-ESO-1 and or LATE1A MRA expression that's positive. And these are patients who have progressed on multiple prior therapies. And here, patients will undergo pre-screening in order to identify the HLA expression as well as tumor antigen expression, after which period of time, if they pre-screen positive, will undergo formal screening, enrollment, and leukapheresis with hopefully short manufacturing process, which is the hope of the study, is identifying a more feasible and streamlined process to get this treatment in quickly for patients who require more effective therapies. And so in summary, we are very looking forward to having this study up and running, which is going to be looking at a best-in-class third-generation TCR-T for the treatment of advanced solid tumors. And again, the idea here is that we're looking at a design where we're looking at a third-generation TCR to increase affinity that's better than affinity mature TCRs. It will incorporate a PD-1/4-1BB co-stimulatory switch protein, which will counteract the tumor microenvironment and removing the need for CD4 positive T cells. We're hoping this will be a much more streamlined process with a shorter manufacturing time, with a 20-day vein-to-vein time, potentially eliminating need for bridging therapy and improving patient outcomes. We're hoping for an improved product quality, that it will increase stemness and clinical efficacy. And then obviously, hopefully that with the results of the EPITOME 1015 study, we can further increase the availability of TCR-T therapies for this patient population. And with this, I will hand it over to Dr. Crame for introducing the pipeline.
Thank you, David. Now, you've heard David speak in depth about our most advanced and clinic-ready program, Medigene 1015. And I'd like to reiterate Selwyn's words here with this slide demonstrating Medigene's exciting, broad and expanding pipeline, leveraging our 3S TCRs in multiple TCR-guided modalities, which all benefit from the precise delivery and specific targeting of tumours, with the aim to get rid of this disease in a way that is safe and tolerable for our patients. Now, the clinical data for Medigene 1015 and our partner programs will serve as validation for our extraordinary TCRs across modalities. In 2025, we will have multiple catalysts with near-term inflection points, with Medigene 1015 clinical data being generated throughout the year. That's just one of the examples of what is to come. And of course, being head of clinical development, a program that is close to my heart. And lastly, over the past year and a half, Medigene has delivered and exceeded on promises made regarding our end-to-end platform expansion, our pipeline expansion, and advancement, including the multiple TCR-guided off-the-shelf modalities, as well as expanding on existing and building new partnerships with both academia and pharma. Now, with a large amount of progress still under wraps, I look forward to updating you in the coming months and years. And this concludes our presentation for today.